Hey, I'm Dr. Oliver, and this is my video on cellular respiration. When we talk about cellular respiration, we're talking about a series of chemical events that occur within a cell. So to get oriented, let's say this circle represents the area of a cell, and within the cell we're going to have various organelles, such as a nucleus, the rough endoplasmic reticulum, smooth endoplasmic reticulum, the Golgi apparatus with a bunch of vesicles, some cytoskeleton, a couple of centrioles in case this cell wants to replicate, and of course, mitochondria. This is the organelle of interest in our talk on cellular respiration, so let's take a closer look at this organelle. The organelle mitochondrion is going to be found floating in cytosol, which is the fluid matrix of the cell. So this blue area is going to be the cell's cytosol or cytoplasm. Now, if we look at the structure of the mitochondrion, we're going to see that it has an outer membrane, an inner membrane, a region in between the two membranes called the intermembrane space, and a fluid-filled compartment called the matrix. Now here we have a close-up of the mitochondrion membrane within the cytosol. The blue region is the cytosol or the cytoplasm, and here we have the outer membrane of the mitochondrion, and here we have the inner membrane. The dark area in between is the intermembrane space, and this lower area is the matrix, the compartment within the mitochondrion. Now, in order to fully understand what's occurring during cellular respiration, we need to know where metabolic events are occurring. Some are going to occur in the cytosol, some will occur in the matrix, and some will occur at the inner membrane and in the intermembrane space. The first step in cellular respiration is glycolysis, and this process occurs outside of the mitochondrion in the cytosol. So what is glycolysis? Well, glycolysis is the breakdown of glucose. And we're going to track the metabolism of one molecule of glucose and see how many ATP molecules we get because, well, making ATP is the purpose of cellular respiration. So when glucose undergoes glycolysis, it's going to require a couple of ATP molecules to energize the reaction and to get it to move forward. Once the reaction is complete, our products are going to be 2 pyruvate molecules, 4 molecules of ATP, and 2 molecules of NADH. Now, because we used 2 ATP molecules to get us going, the glycolysis reaction is said to net 2 ATP despite having made 4. Glycolysis is the only process of cellular respiration that does require ATP, and that might be good to know. The other important thing to know about glycolysis is that it does not require oxygen. It can occur in the presence of oxygen, but it doesn't need it in order for the reaction to occur. In fact, glycolysis is the only process of cellular respiration that does not require oxygen. The rest of the reactions do, and that may be good information to know as well. Now that we've completed glycolysis, what's going to happen next? Well, the ATP will run off to energize something somewhere else, and the 2 pyruvates will be shuttled into the matrix of the mitochondrion for further metabolizing, and the 2 NADHs will also be moved inside of the mitochondrion. Now we're in the matrix of the mitochondrion, and pyruvate is going to undergo the next step in cellular respiration, which is pyruvate oxidation. So our 2 pyruvates will combine with oxygen and coenzyme A to produce 2 acetyl-CoA molecules, 2 more NADHs, and 2 molecules of carbon dioxide. The carbon dioxides will be eliminated as a waste product. And we're going to tuck these 2 NADHs over to the side, and we're going to move the 2 acetyl-CoA molecules out of the way to make room for the next step. The next set of reactions to occur in cellular respiration make up the citric acid cycle, and this has also taken place in the mitochondrial matrix. So we're going to take one of these molecules of acetyl-CoA, and we're going to add it to a molecule that's called oxaloacetate or oxaloacetic acid, which is a 4-carbon molecule, and this is going to create citric acid or citrate. And this is going to be a 6-carbon molecule. Now, citrate will reduce NAD to NADH, and a carbon is going to be cleaved off of citrate, and this carbon is going to combine with oxygen to create a molecule of carbon dioxide. So we're going to be using oxygen in the citric acid cycle. And we're also going to produce a molecule of ketoglutarate, and this is an intermediate product in this process. Now, ketoglutarate will reduce another molecule of NAD to NADH, and another carbon is going to be cleaved off, creating another molecule of carbon dioxide, as well as another intermediate molecule, which is called succinate. Now, at this point in the cycle, we're also going to produce a molecule of ATP. Now, succinate will reduce a molecule of FAD to FADH2 and is going to create fumarate in the process. And fumarate will reduce a molecule of NAD to NADH to create oxaloacetate or oxaloacetic acid, which is that 4-carbon molecule that we started with. Now, because we have 2 molecules of acetyl-CoA to metabolize, we're going to run this cycle for a second time. And with 2 turns of the citric acid cycle, we're going to generate 4 carbon dioxide molecules, 6 NAD molecules, 2 molecules of FADH2, and 2 molecules of ATP. And we're going to put those NADHs and FADHs to the side along with the others because we're going to be dealing with those molecules in just a minute. So we've taken a couple of spins around the citric acid cycle, and now we're ready to perform the next set of reactions, which involves oxidative phosphorylation. Now, this reaction involves the intermembrane space, which is going to be populated with a bunch of hydrogen ions, as well as various transport proteins which are embedded in the inner membrane of our mitochondrion, such as this ATP synthase. Now, ATP synthase is going to move hydrogen ions from the intermembrane space into the mitochondrial matrix. And when this occurs, it triggers enzymes to take an ADP and a phosphate ion and combine them to make a molecule of ATP. Now, it takes 3 hydrogen ions moving through the ATP synthase to trigger this reaction. So every time 3 hydrogen ions get moved into the matrix via ATP synthase, an ATP gets created. Now, ATP creation is great, but we're ending up with all of these hydrogen ions inside of the mitochondrion, which can create a problem as this is going to alter the pH of the matrix. So this has to be addressed. But not to worry, there is a remedy, and this remedy is the electron transport chain. So what's that? Well, the electron transport chain is a series of transport proteins embedded in the inner membrane of the mitochondrion, and these proteins are going to move these hydrogen ions back into the inner membrane space. So how does that happen? You know all of these NADHs and FADHs we've been stockpiling? Well, now's the time that we're going to use them. And we're going to take one of these NADH molecules and add the transport protein you see right here. It's going to oxidize into NAD, which is going to release a couple of high energy electrons. Now, this transport protein is going to use the energy of this reaction, and it's going to move 2 hydrogen ions from the matrix back into the intermembrane space. Now, the electron transport chain is a fairly complicated set of reactions, and I'm trying to keep these concepts fairly general. So without going into a great deal of painful detail, I'm going to say that it's at the transport proteins of the electron transport chain that we're going to use all of the NADH and FADH2 molecules, and I'm going to leave it at that. Now, if you're taking a general biology course or just looking for a refresher on this topic, this explanation should be enough. If you need more than this, let me know, and I'll make another video in more detail. Painful detail. Next, the electrons that we just liberated in the oxidation of NADH to NAD will be shuttled to the next transport protein in our transport chain. But because we've used some of the energy released when they were previously created, they're not going to be nearly as energetic. However, they're still going to be used in another reaction that will move two more hydrogen ions out of the matrix and back into the intermembrane space. And then after that, these electrons will be shuttled to the third transport protein, where another reaction will occur, moving four more hydrogens from the matrix into the intermembrane space. And see there, we're cleaning house and the matrix is looking better already. Now these tired old electrons still have one more job to do, and at this point they're going to combine with a couple of hydrogen ions and oxygen to create a water molecule. And there you have it, cellular respiration. So we've been through a lot of processes and we've accumulated a lot of these NADHs and FADHs and then used them in the electron transport chain. So what's the bottom line with these molecules? What's the real deal here? First, let's remind ourselves from where we got all of these NADHs and FADHs. During glycolysis, we produced two NADH molecules. During pyruvate oxidation, we made another two. During the citric acid cycle, we made a total of 6 molecules of NADH, and when we add these up, we have a total of 10 NADH molecules. Now also with the citric acid cycle, we made a couple of FADH2 molecules. So now we need to answer the question, why are these molecules so important? Well, it's because the oxidation of these molecules creates the energy that the electron transport chain uses to move the hydrogen ions out of the matrix back into the intermembrane space, where those hydrogen ions will be made available to ATP synthase to make ATP. And this means that NADH and FADH indirectly contribute to the production of ATP, and that's why these two molecules are important. So now that we know all of that, we know that each molecule of NADH moves enough hydrogen to create 3 molecules of ATP, and that each FADH molecule will lead to the creation of 2 ATP molecules. And as we just saw, from beginning to end of cellular respiration, we create 10 NADH molecules, which means that this will lead to the making of 30 ATP. And for our 2 FADH molecules, this will lead to the creation of 4 ATP. Now that's a lot of ATP. In fact, most of the ATP that's made in cellular respiration is made by converting NADH and FADH into energy. Most, but not all. Keep in mind that we also made a couple of ATP during glycolysis as well as during the citric acid cycle. When we add all of these together, we can see that from one glucose molecule, we get a total of 38 ATP molecules when metabolized through the processes of cellular respiration. So hopefully at this point you have a good understanding of cellular respiration, and if you do, this chart I'm about to present is going to be a gift because it summarizes everything that's happening on one easy to read page. So first off, cellular respiration can be broken down into 4 sets of reactions: glycolysis, pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation. And when we think about any reaction, we can describe them in terms of inputs, that is, the things that you need to make the reaction happen, and outputs, the things that you end up with as a result of the reaction. Now with glycolysis, our inputs are going to be glucose and a couple of ATP molecules. Our outputs are going to be 2 pyruvate molecules, 4 ATP molecules, and 2 molecules of NADH. Now this reaction is going to net only 2 ATP because we're going to use 2 ATP to get the reaction going. And remember, of the 4 sets of reactions in cellular respiration, glycolysis is the only one that requires ATP to move forward. Now with pyruvate oxidation, our inputs are going to be those 2 pyruvates from glycolysis combined with coenzyme A and oxygen. And our outputs are going to be 2 acetyl-CoA molecules, 2 molecules of NADH, and 2 carbon dioxide molecules. With the citric acid cycle, our inputs are going to be those 2 acetyl-CoA molecules from pyruvate oxidation, as well as some oxygen. And our outputs are going to be 2 ATPs, 6 molecules of NADH, 2 of FADH, and 4 carbon dioxide molecules. And lastly, with oxidative phosphorylation, our inputs are going to be all of the NADH made from the previous 3 reactions, which is 10 NADH molecules, along with the 2 FADH molecules made from the citric acid cycle, as well as some oxygen. And our outputs are going to be about 34 ATP molecules and water. Now there's a couple more pieces of info I want to include here, and these pieces of information can be divided with a line right there. Now the first piece of information concerns where these reactions take place. Well, glycolysis occurs in the cytosol, and the other 3 reactions occur in the mitochondrion. And that's good information to know. The second piece of information is which of these reactions requires oxygen. Glycolysis does not require oxygen. In fact, it is the only reaction in cellular respiration that does not require oxygen, and the other 3 reactions do. And with that, this page pretty much gives you a complete picture of what's happening during cellular respiration. Now, I'm kind of stalling so you can get a screenshot and print it out, or memorize it, or whatever it is you do to get that A. So that's about it. Hey, thanks for watching. Be sure to like up this video and subscribe to my channel. And if there's a topic you'd like me to make a video on, leave that in the comments. And as always, do really well on your next exam. And we'll see you in the next video.